This is G protein coupled receptors and membrane signal transduction by Clayton Kline. Okay, the first step in G protein coupled receptors is there's a ligand and it binds to this metabotropic G protein coupled receptor. When the ligand binds, it causes a conformational change in the heterotrimeric G protein. It's a heterotrimeric because it's made up in alpha, beta, and gamma subunit. It also has a bound molecule of GDP. The conformational or shape change of the G protein receptor causes the alpha subunit to displace the bound GDP molecule. So this GDP molecule is going to leave, and these GTP molecules are going to come in and replace it. GTP binding to this alpha subunit causes it to displace from the beta and the gamma subunit of this trimeric protein. So this alpha subunit, along with the bound GDP, is going to come over here and bind. Draw a little binding site. And we have a bound GTP. Marker roll. And it's going to activate this enzyme called adenylyl cyclase. Activation of this adenylyl cyclase enzyme will convert ATP to cyclic AMP, which, in case you're wondering, is our second messenger. This is what signifies metabotropic receptors and differentiates them from ionotropic receptors. Ionotropic receptors use ion channels, and G protein metabotropic coupled receptors uses second messenger. So cyclic AMP will then bind to protein kinase A, which is over here, and it will bind to the regulatory units of this protein kinase A, causing a separation of the catalytic units, which is signified by the pink squares, and the regulatory units are signified by the orange squares. The cyclic AMP will bind to the regulatory squares of the protein kinase A. The catalytic units of this protein kinase A, which is again signified by the pink molecules, will then be able to disassociate from the regulatory molecules and come down to this molecule called phosphorylase. In doing so, will cause phosphorylase and will cause activation of phosphorylase, which will then be able to bind to what this is over here is glycogen. Binding to glycogen will then be able to separate these glycogen molecules and convert it to glucose, which will then be able to be released by the cells. A phosphodiesterase at the end cleaves cyclic AMP, and the cleavage of the cyclic AMP is activated by a phosphorylation catalyzed by protein kinase A. So just to review, we have a receptor, we have a transduction, and we have a secondary messenger, which then leads to a target cell. And turning off this will be the cleavage of the cyclic AMP, and cleavage of the cyclic AMP will shut down this and will bind the alpha subunit back to the beta and gamma subunit. How many minutes?